Hello, I'm Steve Matsuoka. I work with the USGS Alaska Science Center, and it's my great pleasure to be with you today to speak about the demographic drivers of population change in eastern versus western populations of rusty blackbirds. Luke Powell was originally scheduled to give this talk, but could not, so I'm here on Luke's behalf. Clark Rushing is the lead author and the mastermind behind the models and the numbers that I'll be presenting with you today. I have a long list of very capable co-authors, one of which is the late Russ Greenberg, who we know as the godfather of rusty blackbird research and conservation. His work on this began in 1999 with his paper with Sam Droege in Conservation Biology that sent out the warning signal for the species population decline. In 2005, he founded the International Rusty Blackbird Working Group and chaired the group until 2012. During this time, he helped initiate studies throughout the species range, including a collection of breeding studies that were done on breeding demographics. This included the Acadia forests of New England, where Carol Foss and her colleagues led up a collection of studies. And on the opposite end of the breeding range in Alaska's boreal forest, where Dave Tesler and his colleagues led up a collection of studies. The Alaska population is estimated to have a little over 900,000 birds based on Partners in Flight's estimates. And Jay Wright recently estimated that the migration distance is about 5,000 kilometers for this population. Out east, we see a genetically distinct population in New England and the Maritime provinces, a much smaller population of about 26,000 birds estimated by PIF, and a much shorter migration. In Russ's 2010 commentary in The Condor, he laid out his vision for looking at demographic processes and linking these to population growth in the species. He recognized how population limitation could occur during any point of the annual cycle through deficits in vital rates. In this presentation, we have three objectives, all following up on Russ's vision for analyzing these data. The first was to estimate and then compare vital rates between these two populations. Second, we looked at the annual vital rate estimates to see how they correspond with changes in abundance. And lastly, we simulated how vital rates might be increased with an eye towards increasing population sizes. Again, the data I spoke of earlier were collected over a 10-year period. There are four different types of data that we're analyzing here, and the years of which those data were collected in each one of the regions or both. We have telemetry data collected 4 years in the eastern population. One of those years included data on post-fledgling survival, and then all four are for adult breeding survival. Both adults and young were banded and subsequently resighted in both populations most years. We have nest monitoring data most years and breeding bird indices of abundance in both populations in all years. We fit these data to an integrated population model that models abundance as an autoregressive function of vital rates. We implemented this in a Bayesian framework where we're jointly estimating the growth rate and the vital rates through a collection of integrated models. First, we analyze the abundance using a state space model applied to BBS data. We estimated fecundity, or the number of offspring produced per female using Poisson GLM. We estimated apparent survival for both adults and juveniles using color band resighting data in a Cormac Jolly Seabird model. For the Alaska population, we estimated breeding season survival for adults by resighting adults several times over the breeding season. And in the East, we estimated this using telemetry data and a known fate model. The model has 2 seasons, a breeding season and a non-breeding season. The model estimates female population size as a sum of the age-specific population sizes. The first being the hatching year population size, which is a product of the sex ratio, fecundity, and the number of adult females in the population. Second year population in a given point in time is a function of the number of offspring raised the previous year and their seasonal survival probabilities. And lastly, the after second year population, which is a function of the number of adults in the population the previous year and their survival probabilities over the intervening period. Here are the mean vital rate estimates in separate columns for East and West. And so I'll draw your attention to the adult non-breeding season survival, which is about 10% higher in the East compared to the West, which is in keeping with what we might predict based on the much shorter migration distance for the Eastern population. But we see the opposite for juvenile non-breeding survival, where the East is much lower than in the West. And it's possibly suggestive of a vital rate deficit here for the Eastern population. Annual estimates of survival are similar, higher in the East than in the West for adults, but lower in the East than in the West for juveniles. Population growth rate is lower in the Eastern population, suggestive of about a 3.3% decline per year, where out West is relatively stable. This figure shows the annual estimates of abundance and the vital rates. Here I'll first draw your attention to the black dots, which are the annual estimates of abundance, showing a decline over the course of the study. If we look at these periods of decline from like 2011 on, we see that the decline in abundance corresponds with these decreases in survival during the non-breeding season, particularly for juveniles. Out West, we see that the population is more stable again, and that periods of decreasing abundance, like we see here from 2009 to 10, correspond with low fecundity and low adult survivorship during the non-breeding season. So for both populations, it looks like survival during the non-breeding season is particularly influential on abundance. Next, we ran simulations of population change using our integrated population model. And here we asked the question of how vital rates might be increased in order to effectively increase population sizes. In these simulations, we use the main vital rates and use them to project future population sizes at annual steps over a 20-year period. We kept all the vital rates at their mean rates except for one, which we increased by 2, 6, or 10% in each one of the simulations. Here are the results from our simulations of increasing fecundity. The y-axis is abundance. The x-axis is the year of the simulation. The black dots are the observed abundances from the breeding bird survey from years 1 to 10. And then the lines thereafter are the simulations. The dashed line are the vital rates held at their mean levels unchanged. You see a 29% increase from year 10 to year 30 in the simulation. The green lines of different shades, of darkening shades, are increases in fecundity from 2 to 10%. So we see as fecundity is incrementally increased, that the population size increases in response. Out west, we see that when the vital rates are unchanged from their mean values, that the population is projected to decline by about 50% over 20 years. Under the two scenarios of 2 and 6% increases in fecundity, the population remains in decline. And not until we increase fecundity by 10% do we see the population increase in numbers over the simulation. This figure shows all the vital rates and scenarios. So each panel is a different scenario of increasing vital rates. So we see under the 2 and 6% scenarios that the population is increased the most by increasing non-breeding season survival. I'll point out in these simulations that we increase survival in both adults and juveniles, and so that in part explains the larger impact of increasing non-breeding survival in our simulations. In the 10% scenario, we did not run the non-breeding season survival here, since the preliminary runs led to unrealistic increases in population size. But we see that increases in fecundity, post-fledgling survival, and summer survival all have similar impacts on population size here. These are results for the simulations in the West. So in these simulations, under all scenarios, the population remains in decline, except for when we increase non-breeding survival by 6%, and when we increase fecundity or post-fledgling survival by 10%. So our conclusions, all arrows seem to point primarily to the non-breeding season. We see in the eastern population that juvenile survival during the non-breeding season is low and possibly in deficit. Declines in breeding abundance seem to correspond with periods of low survival during the non-breeding season in both populations. And recovery efforts appear to be most efficient if they target increasing survival during the non-breeding season. And I'll end on a somber note. Over the course of our research, we lost 3 of our dear collaborators. I spoke of Russ Greenberg earlier. But Bill Glanz, a university professor at the University of Maine, passed away in 2014, and Dave Tesler, our dear collaborator up here in Alaska with the Alaska Department of Fish and Game, left us in 2017. So I thank you for your attention, and I hope to catch up with you during the discussion period.